Okay, let's go ahead and get started. Couple of quick announcements. Again, mea maxima culpa for last time, thanks to Portland Public Schools and their buses, as far as me being a little late then. I noticed that since I very quickly tried to get everything up and running last time, that I didn't manage to get an audio recording. I have this wonderful video recording of all the slides, which is kind of worthless. So what I will do is hopefully over the weekend, unless somebody has an extra audio copy, over the weekend I can go back and overdub it and hopefully say similar things to what I said the first time round. This time everything seems to actually be working properly, so I will go ahead and post that. Another thing from last time is I didn't count clickers, again, just because of all the setup and things. Clickers as of today will count. I've had a couple of people ask about clickers and sort of the whole, are they being registered, et cetera. What I'm going to try and do is, after class today, is upload all of those scores to D2L. And that way you should be able to know if you're connected, et cetera, with all those appropriate numbers. I don't expect you to be registered until the midterm. But if you have any questions about that, probably the best thing to do is to email me. You can also email me your clicker number if there are some issues with that, and we can try and input all of those. So any questions about that aspect of things? No, the next slide is not a clicker question. Okay, so starting out where we left off last time with Martha Chase and Al Hershey and their experiments with the T-even bacteriophages to basically repeat the work in a very different system with what Avery had done already in terms of purifying the transforming principle. So what they did, couple of things here that I wanted to point out. I don't expect you to remember exact dates, but just here, this is 1952. We'll talk about the Watson and Crick paper in 1953. Basically, these things are happening all at almost exactly the same time, and not that long ago. So what, well, basically, Martha Chase did and Al Hershey said to do was to label DNA and protein differentially with each other. Protein labeled with S35. That sulfur comes from where in proteins? Cysteines and methionines. So the only place that you'll have any kind of radioactive label is going to be in the protein if you use sulfur. What about P32? So phosphorus, that's actually in their DNA to a great extent, but you also have it in proteins too. So there's going to be a little bit of background from that. Fortunately, in bacteriophage, these bacterial viruses, not very much. But there still are some cases of that. So that's not as complete a labeling process. And two different preparations. So one preparation of bacteriophage with S35, the other with P32. Another aspect about the year here, this is soon after the Second World War when nuclear power, nuclear bombs, et cetera, are happening. So there's actually a lot more availability of radionucleotides. Basically didn't exist before this. So these kinds of experiments couldn't have been done any earlier than that. Then what they did was infect cells, again, 2 different times. Actually, it's about 7 different times if you want the paper. Take my molecular genetics course next term. We'll talk about the whole thing. Separate infections with these different viruses, separate binding, killing the viruses, et cetera. But the main thing is do your infection, these 2 different preps, 2 different times. Separate the virus particles from the cells. This is your E. coli smoothie. There's basically a blender that was used to do the experiments. You can actually see these in some labs. And then basically, where's your radioactivity? Is the P32 in the cells and the viruses of the next generation? Or is it the S35 that's in the viruses and cells in the next generation? So really beautiful experiment, the way they set it up. And basically, this is the idea. Unfortunately, this is a little oversimplified here because it's actually showing 2 different labeling in the bacteriophage, the virus at the same time. It's actually 2 separate experiments that they did. They did one where you had labeling of the DNA with P32, one with labeling of the protein with S35. You put these together with the E. coli. The genetic material comes inside the cell. You remove the virus particles, again, the blender, and then you look to see what is being transferred. Is it the P32 that's being transferred, or is it the S35 that's being transferred? And through that, you can say, okay, if it's P32 being transferred, it's your nucleic acid, probably, because the vast majority of what's in there is now going to be your nucleic acid, or the proteins for the S35. Is everybody clear on this? Great, so you can all answer clicker questions on it. So clicker question 5.1, because this actually starts to count. If Chase and Hershey, oops, get this to actually start, had found, yes, base frequency code. I know it is, sorry. Chase and Hershey had found that S35 was passed from the old virus to new virus in the experiment. They would have concluded that DNA was the genetic material, RNA was the genetic material, protein was the genetic material, that had contamination. The viruses contained protein, or the undergrad who was doing the project, had no clue. Sorry, we don't have F. Five, 3, 2, last chance, go. Few people seem to think that DNA was the genetic material. I would have trouble finding a sulfur atom in here. There are lots of problems with these DNA models, by the way. If you want to come up and check them out later, it's part of the reason why I brought them. But that would be that protein was ejected. That's the whole idea. Is S35 is just labeling proteins. So if you saw that in the next generation, that's what's being passed along. And so that's what they would have concluded in that particular case, which 93% of you have, so it looks like we're in pretty good shape. Okay. Get rid of that. So at this point, again, very soon after this experiment was published, Watson and Crick, doing actually Crick and Watson from left to right here, started to do a lot of model building, which was actually the opposite of what Crick was supposed to be doing. He was actually supposed to be working on proteins at the time, and Jim Watson, who was this upstart postdoc from the United States, the way he behaves personally is not unlike our current president. Not such a good thing. Still, that's all we'll say about that. But it was interesting that Jim Watson was actually a postdoc at the time, but was considerably younger than Francis Crick, who was the graduate student at the time, partly because this was in England. Behind them is Christchurch. This is the cathedral in King's College. And if you listen to the carols at King's College, that was actually recorded in that particular building there. So they basically got together and were looking at trying to come up with a model for the structure of DNA. What did they know? They actually knew a whole bunch at the time. We talked a little bit about this last time. They knew the structures of the nucleotides, which we'll talk about here in just a second. They knew that the linkage was a 5' to 3' linkage, and we'll talk about 5' and 3' in just a second here. Phosphodiester bonds. I probably should have put quotes around Chargaff's rule, and that was that the concentration of adenines and thymines was about the same, and guanines and cytosines was about the same, not actually identical in Chargaff's measurements. But again, we should have a quote on the rules there. What did they steal? Probably stole. There are a couple of books about this. One is The Double Helix, which is Watson's account of it. There's also a really nice biography of Rosalind Franklin. And it is probably pretty clear that they stole these pictures. And in fact, even in Watson's book, they mention this. So what do they have? Basically, what they had was an X-ray diffraction image. In fact, two different kinds of DNA. There's a so-called A-form DNA, which is what was up here, and the B-form DNA down here. And fiber diffraction is actually a very interesting kind of technique. Basically, what you're doing is you're shining X-rays down the long length of whatever it is that you're looking at. And if everything is arranged in a nice symmetrical form, you'll get reflections. And those reflections are what ends up on this photographic plate. And what they saw there was that it was about 2 nanometers in diameter, clearly helical, and it had repeats at 0.34 and 3.4 nanometers. And that's what you can see. We'll look at the breakdown of this in just a second here. They also had Linus Pauling's helical model for DNA. Now, Linus Pauling, as we talked about before, Portland's probably most famous son, alpha helix, beta pleated sheets, hydrogen bonds, absolutely brilliant guy. He was also working on trying to figure out what the structure of DNA was. So here this is. This should, again, hopefully be a review. We talked about it a little bit before, but wanted to go over this in a little bit more detail. And the main thing here is really all about 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, and really 3 prime and 5 prime. We're going to spend probably ad nauseum talking about 3 prime and 5 prime here. And this is just to orient you where 3 prime and 5 prime are. This has to do with the ribose, which is in the backbone, and it's just numbered from where the base is hanging off of. The base always comes off the 1 prime position. 3 prime position has an OH. The 5 prime position has a CH2OH. And you always have a linkage to phosphates at 5 prime and at 3 prime. Arnold Levine, who said that protein was the genetic material, had figured out this is a 2 prime deoxyribose, which means that there's no OH here. That's, of course, the big difference with ribose, is because it actually has an OH at that position. Some people have asked me, and you may wonder why all the primes here rather than actual numbers. And that's because the purines and pyrimidines themselves also have numbering on them. I don't expect you to remember the numbering on these. I always get confused as far as that's concerned as well. But that's why these are always prime. So they also knew the structures of the individual nucleotides, adenine, guanine, which are the purines. The way I remember this is short name, big molecule. And the pyrimidines, which are the long name, smaller molecule, cytosine and thymine. These are the data that they begged, borrowed, stole from Rosalind Franklin. A couple of things that you can get off of this. The first one is that there's this X pattern. And actually, it's not just an X pattern. There's a secondary X that goes up here. It's a little harder to see in this image. But that tells you that this fiber actually has helical repeats in it. And so it's that X structure which gives you a helix. And all of these so-called layer lines here on the side are telling you where all of those repeats are, the 0.34 and the 3.4 repeats. It's this distance from here to the middle which gives you the 2 nanometer repeat or the 20 angstrom, diameter of your helix that's there. And this is the model from their paper on the outside. This is just basically a quick reminder, which I forgot to mention before. There's a really nice section in the textbook in Chapter 2, all these different panels with all of the structures of all the different nucleotides, proteins, etc. So if you want to go back and look at the chemistry, I think it's a really nice review, and it's all the chemistry that we really need as far as this course is concerned. The only other thing to mention on here is that each individual nucleotide has these 1 prime through 5 prime. 5 prime and 3 prime are always where your phosphate are, but then you're always in a linear chain. So here, 3 prime end of the chain, 5 prime end of the chain, always linked in the same way relative to each other. So again, this is all stuff that Watson and Crick already knew about. What did they do? They built models. They basically played around with their Legos, although actually it was Legos they got from the machine shop at Cambridge. And put stuff together to try to get it to work. And what they came up with is this model drawn out here, and we'll see the picture in just a second what the real model looks like. Anybody seen the real model or an example of it? There's one in the Smithsonian. It's really pretty amazing. It's about this tall. It's really pretty impressive. And if you have a chance to take a look at it. But what they found was basically a couple of things. One, of course, it was double-stranded. You couldn't get that from any of the data. 2 strands was a new thing. Complementary strands, you couldn't get that from any of the X-ray data. But in order to get the model to work, that you had to have these 2 complementary strands, and they had to be anti-parallel relative to each other. So what's anti-parallel? That means that one of the strands is going from 5' to 3', the other one is going from 3' to 5'. So they're opposite orientations relative to each other. They're right-handed helices. We talked about the right-handed helix before. I've got lots of examples of helices up here again, mostly left, some right. One way I like to think about DNA, if you come down here afterwards with the model, is sort of also a right-armed helix. So I can wrap my right arm around here, moving upwards, and it wraps nicely. Try to do the same thing with my left arm, it just bangs into stuff. So another way of just thinking about things. How do you get this kind of structure? Phosphates on the outside and the bases on the inside. This was the big difference between their structure and Linus Pauling structure. Linus Pauling structure, for reasons that are not entirely clear, had all the phosphates on the inside and the bases facing out. And probably the reason for that was that that's where you have the variability. If this is the genetic material, you need to be able to access that genetic material. So it makes sense, but not so much from a chemical point of view. Why does it make sense from a chemical point of view? Because they're all negative charges. You're putting all these negative charges right next to each other. That really doesn't make sense. And then, so with the bases on the inside, which was probably the real breakthrough that they made here, bases on the inside, they can then interact with each other with hydrogen bonds, which is what Linus Pauling had already come out with, and perpendicular to the, if you think about it, vertical kinds of anti-parallel strands that you had on the outside. A couple of things they had to do about this, C4 and C6 of cytosine and adenine relative to each other. Again, I don't expect you to remember these numbers. But basically, they're amino groups, not amine groups, so they're NH2. And that was really necessary in order to get those hydrogen bonds to form. Same thing is true of the ketose, C6 of guanine and C4 of thymine. Because of this structure, one of the things that you end up with is major and minor grooves in the DNA. And we'll spend a while talking about that because it's actually very important in terms of interactions of DNA with other proteins and seeing what the actual sequence is of that particular piece of DNA. So right-handed, left-handed helices, down here on the bottom. Some people like thumbs, point up the way your fingers wrap. My fingers don't wrap that way. But a number of different ways. Another way you can think about, yeah. Where are you? Oh, it's here. Yeah.
On the previous slide,
Yeah.
you were saying that the amino group, the NH2,
Mhm.
is necessary for the hydrogen bonds
Yeah.
of cytosine and adenine, and then the ketose are for the guanine and thymine.
Correct.
Okay.
So that's actually basically right here. sorry, I missed your question. And by the way, if anybody, I don't notice a hand, yell. Yeah, so right-handed, left-handed down here. Another way to look at right and left-handed helices is if you just grab one and look at the way the helix is going away from you. If it's going clockwise, it's right-handed. If it's going anti-clockwise, it's left-handed. So these are just other ways of looking at right and left-handed helices. B-form DNA is a right-handed helix. There actually are left-handed helical forms of DNA that we may have a chance to talk about a little bit later on. Getting to your question about the ketose and the amino groups, that's up here. Here, with cytosine, if it were an imino, I would have a double bond here, then you'd have different hydrogen bonding interactions here. Whereas here, the H bond, or so this hydrogen here, can form a bond, in this case to the keto group, which is on the guanine. And if you flip these around, do amino for cytosine and an enol for guanine, the structure doesn't work. And that's actually one of the things that Watson points out in his book, is it was really critical to know those particular things there. So this is an image of that particular model right here. A couple of things to note on the model. One is that these base pairs are actually offset from the middle. They're not actually straight down the middle, which is one of the problems with these two models that I have over here. And the other one, how many people are old enough to recognize this reference down here? It's only a model. Sometimes I show that video, but we won't do it this time. So here, this was, in fact, interestingly enough, one of the mistakes that Watson and Crick made when they came up with their model. It's a little, I don't think you can actually see it on here. If you look carefully, I don't see it. So if you look between the G's and the C's here, how many hydrogen bonds do you see? And A's and T's? Curious. But what do we know now? That's actually not the case. So G's and C's actually form 3 versus 2. And there's been a lot of discussion about this, and apparently Crick's idea was that you can't have it be different. They all have to be the same. And so therefore we're going to have 2 hydrogen bonds rather than these 3 hydrogen bonds between the 2. But again, it's really critical here that we have our amino here, keto here, so you can have this hydrogen bond interaction here. Here, hydrogen bond interactions again, amino here, keto here, amino here, keto here. And it doesn't work in terms of the structures. Another thing that was really important in terms of putting their model together, you remember that they were perpendicular to the sugar phosphate backbone. If they're perpendicular, what that means is that the positions of what's called the glycosidic bond, why is it called the glycosidic bond? It's the sugar, the glyco, is in exactly the same place here, whether you have a GC base pair or if you have a CG base pair, the position of this sugar doesn't change at all. Same thing is true with A and T and T and A. So you can have the backbone be in exactly the same place for every single one of those base pairs. And that was why the model was so powerful, because it didn't matter what you had on the inside. You still had the same structure in that particular model. So in years past, I've asked people to draw base pairs on midterms. I don't do that anymore. But that being said, I do expect you to know the atoms on base pairs. So if I were to write up a base pair that was wrong, I would expect you to tell me what was wrong with that particular base pair. The other thing that I forgot to mention as we went through, the Watson and Crick paper, how many of you have actually read it already? How many of you are going to have read it by the midterm? There will be a question on that particular paper on the midterm. So just one. So one question that is not going to be on the midterm, however, is our next clicker question, which is here. Crick and Watson's model for B-form DNA had blank hydrogen bonds between guanine and cytosine, including the sugars and phosphates, 2, 3, 4, 8, or 10. Yes, feel free to discuss how obnoxious Stedman's questions are. Five, 4, 3, 2, lift off. Okay, so yes, hopefully everyone agrees. Yeah, so the problem with, or one of the problems with Watson and Crick's structure, we'll talk more about the other problems later, is that they just had 2 hydrogen bonds between guanines and cytosines, not 3, which is the correct case. What about other hydrogen bonds? Hydrogen bonds between phosphates and sugars, et cetera? Are there any? No, there aren't any, fortunately. Okay, so yes, the answer is A. Here, so to risk beating the dead horse here, couple things about the polarity of your double helix. And again, it's an anti-parallel double helix. So you've got a 3' end that's always paired with a 5' end. So the G in one case is going to be paired with a C in the opposite case. This is another way of looking at it here. 3' end here, 5' end here. So the structure only works if you have these in the anti-parallel case. So this is the polarity issue. Another way of looking at it is here. This is your, sorry, I keep trying to use the wrong one here. Here's a 3' end to a 5' end, a 5' end to a 3' end. And here it's quite well shown the difference between the minor and the major groove. And this is true also in the space-filling model. And if you remember back to when we talked about space-filling models, these are the van der Waals radii of each of the atoms. And so if you're really interested in what's interacting with whatever else is there, you really need to use space-filling models to look at it, not models that look like this. It's a lot better way to look at the space-filling model if you really care about the interactions that are happening between the two here. So one of the things, actually, this model's kind of falling apart. I'll use this one. Well, they're kind of both falling apart. Major groove versus minor groove. And you can come up and take a look at this a little bit later on. But here you'll notice the two phosphates are much closer together than they are over here. And so this is your minor groove. That's your major groove. And that just comes out, again, of the model building process that gets put together. I don't have it right now on my phone. There's actually a pretty neat little free app, at least for iPhones, called Molecules, where you can actually download and look at some B-form DNA and play around with it, spin it around, and different structures, et cetera. So major groove, minor groove, again, to risk going over this in too much detail, here's our major groove, here's our minor groove, and the arrows here, again, are supposed to indicate the differential polarity, 5', 3', 3', 5' relative to each other. Mentioned right at the beginning when we were looking at the data that were begged, borrowed, stolen from Rosalind Franklin, there were two forms. There were two pictures that she had, an A-form DNA and a B-form DNA. A-form also has these anti-parallel strands relative to each other. It's also, excuse me, double helical, right-handed helix, but the base pairs are really skewed relative to the rest of the structure. Also has a much narrower minor groove and a much bigger major groove. How do they get, A-form DNA and B-form DNA? It has to do actually with the amount of water which is present in the structure. If you have really dehydrated DNA, you end up with A-form, and hydrated DNA is B-form. The DNA is actually present inside of cells is really neither of these. It's somewhere in between. And as I mentioned before, you also have some cases where you even have left-handed helices that are going to form in your DNA. So it's a great model. It's a great place to start. But that's only the beginning of thinking about a DNA which is there. I keep beating on major and minor grooves. The reason for that is all the information you need to know what sequence of bases you have is all present in the major groove of DNA. The minor groove actually has a lot less information. And so this is diagrammed here. If you look at the major groove in a CG base pair or a TA base pair, all of these atoms that are hanging out in the major groove are really specific for that particular base pair. So here we've got two hydrogens that are not able to form hydrogen bonds, a hydrogen bond donor here in blue, two potential hydrogen bond acceptors, and a hydrogen atom which is not going to be forming any kinds of hydrogen bonds. And just looking at from one side to the other, if you flip this over, now you have a different orientation of all of these individual side chains. If anything, it's almost easier to look at an AT base pair, because one of the ways to tell whether it's a major groove or not is just where is that methyl group. Methyl group is always going to be in the major groove of any right-handed form of DNA. And you come up and look at these structures, it's a big black ball here that's not attached to two yellow ones, which is supposed to be the phosphate. One other way of looking at this is called the DNA recognition code. Again, hydrogen bond donors and acceptors, methyl groups, just the orientation of these particular atoms and how they're going to be interacting with, say, a side chain of an amino acid, you can get sequence-specific information out of the major groove. If you look at the minor groove, on the other hand, between a GC base pair and a CG base pair, it's actually identical. So if you're just looking in the minor groove, and all you can see, because all you can see here is whatever you can interact with, is actually identical whether it's a C on one strand or a G on the other strand, or an A on one strand or a T on the other strand. So for these kinds of sequence-specific interactions, it's all going to be about the major groove. Now there are some, and we'll talk quite a bit about some of the non-sequence-specific interactions, many of which are happening in minor grooves. But yeah, sequence-specific is all happening in the major groove. Any questions about this? And when he says any questions and no one raises their hand, what's he going to do next? Ask you a clicker question, exactly. So most sequence-specific interactions between proteins and DNA disrupt base pair hydrogen bonds, are to the backbone phosphates, are to the backbone ribose, are to base pairs in the major groove, or to base pairs in the minor groove. Five, yes, major groove. He's going to keep banging on the major groove thing. That is definitely true. There are some cases where you have things that disrupt the base pairs. Those are what are called helicases. We'll talk a lot more about those later on. And there are quite a few proteins that are going to interact with backbones, but those are not sequence-specific. Any kind of sequence-specific is going to be interacting with the major groove of DNA. So this doing the clicker questions as we move through is kind of a new thing. I used to do them all at the beginning. Let me know, comments, emails, et cetera, whether you think it's a better or worse thing to do them all at the beginning or later on. So just to finish up with the structure of DNA, one of the things that really amazed me, and I think amazed a lot of the earlier researchers, is that if you've got your nice double-stranded DNA like this, and you pull it apart into the two separate strands, like the helicases I was just talking about, and you let it come back together, it comes back together in just the right way, which is really amazing if you think this is literally thousands, in some cases millions, and even in some cases billions of base pairs of DNA. They find their right partners, and they find their right partners really, really quickly. And that's basically shown here in this particular case. That's what's called a denaturation curve or a renaturation curve. A lot of times people talk about hybridization, just a sec. And what it means is that DNA can be pulled apart into two separate strands, even though they're wrapped around each other, and then put back together in an extremely repeatable, you can do this process, and in this case it's just heating it up, cooling it down. If you heat up your DNA enough, it comes apart. You cool it down, it comes back together, and it's the same thing, and you can do this again and again and again. And it turns out that there's one particular midpoint here that you can get between pulling the two strands apart, being single-stranded versus double-stranded. And it's very specific. Turns out it's almost completely dependent on the percentage of G's and C's that you have in your DNA relative to the percentage of A's and T's, which is another reason why the 3 hydrogen bonds makes a lot of sense. Yeah, your question in the back.
Sorry, my question was just, you mentioned when it goes apart and comes back together, we're going to have some strands that shift for, half of it full, half of it joins, half of it doesn't kind of thing, no?
So the question is, I'm just going to repeat it because it'll probably be hard for people to hear it. Aren't you going to have pieces that will come together in different places and bind there instead? It turns out that that's actually extremely rare unless you have repeated sequences. And so those repeated sequences can then come back together in the wrong place. But in general, you actually come back and find them in exactly the same places. It has a little bit to do with the kinetics of what we call denaturation and renaturation, and we may talk about that right at the end of the course. But as far as we're concerned, you can pull the strands apart and put them back together. They're going to come back in the same place, which is still really incredible. Right in the back first.
So just to clarify, between the G's and C's, you
Yeah.
have 3 hydrogen bonds, which
Yep.
can overcome more thermal energy than the double hydrogen bonds.
Correct. Yes. So
If the G's and C's and the A and T's all had 2 hydrogen bonds, it would come together probably at random and not organize itself correctly.
okay, so I'll try and paraphrase your question here. So the idea with the 3 hydrogen bonds between G's and C's, that gives you more thermal stability than just the 2 hydrogen bonds that you have between A's and T's. That is exactly correct. Now, if you had just 2 hydrogen bonds between A's, T's, G's, and C's, you would still have them come back together in the right way because they can't, A's can't form hydrogen bonds, even 2 of them with C's. So they're always going to come back together in the appropriate orientation. I should do this flipped over to make sure that we got our opposite polarity relative to each other here. So you pull them apart, you put them back together, they're still going to come back in that same place. What's going to be different if you have different A's, T's, and G's and C's, that you have 3 versus 2, is this position where you have half denatured and half pulled apart single strand, half pulled apart in double strand. So if you have more G's and C's, more hydrogen bonds, this TM is going to be over here at a higher temperature. If you have less, it's going to be over here at a lower temperature. Yeah, there's another question down here, I think. Okay, good. Thank you. So that's all we're going to talk about in terms of direct DNA structure. Now we'll start moving away from that just double-stranded Watson-Crick B-form DNA and think about what real DNA looks like. Start out talking about chromosomes, and particularly in terms of karyotypes. This has become old technology, but I think it's really visual, so it's nice to look at. These days, people just sequence the whole darn thing, and it's quicker. Human chromosomes, talk a little bit about the human genome and its content, and then we'll talk about how these chromosomes come together. And one of the big problems with DNA is that it's actually pretty darn big relative to the tiny little cells that it's got to fit into. And one of the nice things I like to talk about is that the human genome, if you took the DNA from one of your cells and just stretched it out like this, it would be slightly taller than me in one cell. So the amount of compaction that you have to get is really amazing. And we know a lot about the structures, very basic structures of DNA here. We know a bit more about the first step of condensation, which is these nucleosomes. Condensation and compaction I'm going to treat reversibly here. Pulling things together, that is made of the histones, and then histones packed together to form what's called the 30 nanometer filament. And that's why most of the DNA in most of our cells is, as long as it's an active kind of form. And we'll look at that in a lot more detail. So first, a little bit of terminology. Chromatin. What's chromatin? Chromatin is just your nucleic acid plus the proteins. And this is, since the proteins had way more diversity and there was at least as many of those as you had of the nucleic acid, that was why people until Avery and Hershey and Chase, I should say Chase and Hershey, showed that it was actually the DNA which was the genetic material rather than the proteins. So DNA plus protein. As I mentioned before, human genome, 2 meters long, but it has to be packed into individual cells. So chromatin has to package the DNA, has to squish it down, but at the same time, you need to be able to get your protein into the major groove of DNA so that it can interact there. Or sometimes you need to be able to pull those two strands apart relative to each other. So chromatin's really interesting because it's got to be able to compact and still make things available at the same time. We'll talk about protection. I'll get to in just a sec. We'll talk about the protection of the DNA in just a second. Some of these models have a number of problems with them because they're falling apart. DNA does exactly the same thing. So chromatin helps with that as well. Also, organization of your DNA, particularly if you have to separate your DNA. Talking about the cell cycle, what happens in terms of segregation of your two chromosomes, and just holding them all together in the right place. So there's a lot of proteins. So it's not just that genetic material. The genetic material has to be compacted, available, and organized. Yes?
Are we talking about more than histones here, other protective proteins as well, or are we just, are histones what we're talking about proteins?
So when I'm talking about chromatin, I'm not just talking about histones at all. So histones are part of chromatin, and by weight, they're actually the largest amount of proteins that you have in there. But there are many what people call non-histone chromatin proteins, and we'll talk a lot about those as well as we move forward here. Just as a quick aside, bacteria have exactly the same problem as do archaea with this whole compaction issue. Yeah, they're small cells. They've got slightly smaller genomes for the most part, but they still need to be compacted. And it turns out that compaction and access is just the same kind of problem, only it doesn't use histones. It uses a bunch of other proteins, and we actually know a lot less about how that functions in bacteria and, for that matter, archaea. So first, looking at the compaction issue, if you just look at DNA by itself, again, most of the time it's in what's called this 30 nanometer fiber, which you can see down here on the right-hand side. That's in your normal interphase happy cell. There's still a lot of proteins that are present here. These are a lot of these non-histone chromatin proteins that we're talking about as well. The least compacted form of chromatin is what we have down here. This is your beads on a string. This is just the nucleosomes. Now, when you think about chromatin, it's more like this, or in a metaphase chromosome, it's like this. So it is about a 10,000-fold compaction between just your B-form DNA, actually even less than this, and one of your metaphase chromosomes. Yes?
Just to clarify that what we're looking at is NA, is the edge of a chromosome?
In A, no. So this is, if you just look at the DNA that's present in a cell that's not undergoing division. So this is not a particular chromosome per se. This is all of those chromosomes mixed around each other, which is really amazing that they managed to keep everything sorted out. Okay, so one of the really nice things about these chromosomes is that they're very specific in terms of their size, shape, and also they call the banding pattern. This is the karyotype, so 22 pairs of autosomes. Each of these has a very specific banding pattern. I have no idea how to read all of these things, but people have been looking at this for numbers of years. They can figure this out. 22 normal pairs, and then hopefully everyone knows our sex chromosomes here. What sex is this particular person? It's going to be male because they've got this, radiator gene containing chromosome over here. All kinds of other things. Too many jokes about the Y chromosome. But clearly women are genetically superior to men because they've got a lot more of the DNA. So notice the X chromosome is much, much bigger here. So this is what people always show you in the textbooks, what they look like. But what they look like when you just take out these, now this is not what we were talking about before, the chromosomes that have been compacted in metaphase. This is now, sorry, the ones that weren't compacted before. This is now compacted in metaphase. This is the only time you can actually see them under a normal microscope. All those previous ones were electron microscopy. So with a light microscope, you can actually see them with a regular light microscope, but only when they're compacted in metaphase. There are a couple of different ways of looking at that. One is with that banding pattern that I just showed you before. The expensive way to do this is to paint your chromosomes. And it turns out that this painting process has everything to do with the fact that when you pull those two strands apart and you let them come back together, they come back in the right place. So the way that you do this particular experiment and give each of those chromosomes a different color is to have a specific DNA sequence. It can actually be a pretty short DNA sequence that has, again, very specific sets of nucleotides that has a fluorescent tag on that particular piece of DNA. And you mix and match all of these different fluorescent tags of different colors. And then if you look at them on an epifluorescence microscope, you can see all of the chromosomes as being slightly different colors relative to each other. But that's only because they have different sequences relative to each other. And the fluorescence is going to give you all of these different things. Your mixture here, now of course, it's the autosomes and sex chromosomes which are in here. And basically, here's chromosome 1. It's hybridized to certain labels that give you this structure. Here's the other copy of chromosome 1 here. Here's two copies of chromosome 13. Here's a copy of chromosome 14. There's the other copy of chromosome 14, et cetera. And so you can line all of these things up relative to each other. Why is this important? In a number of cases, there actually are what are called translocations, where one piece of one chromosome moves to another chromosome. This probably happened a lot in evolution, but also happens a whole bunch in cancer cells. And in one particular case, there's something called the Philadelphia chromosome, which is literally a piece of one chromosome that's moved and hooked itself onto another chromosome. Why do people care about the Philadelphia chromosome? Anybody know? Right? Chronic myelogenous leukemia. Anybody heard about that? Anybody heard about Gleevec? The drug developed by our friend, actually his lab's now down the hill rather than up the hill anymore, Brian Drucker, who will probably get the Nobel Prize at some point for developing this. It's a very specific drug that blocks the activity of a particular protein which is misformed because of this chromosome translocation. So just by looking at the karyotype of someone who has a blood disease, you can determine that there's one drug that will basically cure them. It's just really amazing. Yep, absolutely incredible. Unfortunately, most of them aren't like that. So this is that example here. Here's one of those stains, and here's a clear chromosome translocation. Of course, it's a lot easier to see if you've done this color staining, but good people can detect it this way. The other thing, of course, that we do now is we just sequence the whole darn thing. Yes, the back.
Did the drug fix it by treating the protein after it was formed, or did it fix it by fixing the chromosome?
Okay, so the question is, does Gleevec work by fixing the protein or fixing the chromosome? We wish we had ways to fix chromosomes. That would be really wonderful if we could. And in fact, what Gleevec does is it specifically inhibits the activity of this dysregulated protein that we may have a chance to talk about a little bit later on. It's a protein kinase. So we talked about before, it's putting phosphates onto things. And basically what happens is it's super active in these cancer cells. And it was in the process of the drug design, knowing the structure, that they figured out a way to actually block that activity. So that's what the drug does. Yeah, in the back, and I'll get to you.
What leads to the chromosome shifting places?
What leads to the chromosomes shifting places? If we knew that, that would help us a lot, particularly in terms of some of these cancers. We do not know. But it's interesting that you get this very specific shift, again, in something like the Philadelphia chromosome, where you do have then that specific change that happens in that specific place. But we don't really understand why that is. Yeah?
Is this protein a cause or an effect of the shift?
So is the protein a cause or an effect of the shift? So we'll talk more about this when we talk about coding proteins, but it turns out it's actually becomes a fusion protein. So there's actually two proteins that were normally separate before that become one protein. And so it is going to be because of that shift and these two proteins being brought together that gives it that phenotype, so that nastiness which is going on there. And this will become a little bit more obvious, hopefully, in the next couple of slides here. So fortunately, one of the students who was in this class a few years ago went and worked in the karyotyping lab at OHSU. So these are actual real human karyotypes from up the hill. I would have a huge amount of trouble putting all these things together appropriately and figuring out which chromosome, what the heck is this going, maybe some of the tiny ones, that one I could match up, etc. But these people are really good. They can line them all up appropriately. And now we have one of these genetically superior individuals here. And just by lining all these things up and looking where those bands are, you can really clearly see that there hasn't been anything in terms of a nasty translocation, no need to use Gleevec, etc. On the other hand, if you have chromosomes from cancer cells, it looks really messy. So it's not just that one translocation that has happened. In this case, we've got 5 copies of chromosome 1, no copies whatsoever of chromosome 17, 1 of 16, a lot of stuff that's been moving around, etc. So this, getting back to your question, cause versus effect versus any kinds of these chromosome changes, that's a lot more of a question in general in cancers, is whether there's all these changes that are happening, is it due to the cancer or is it what's causing the cancer? So the whole cause and effect kind of thing. It's probably some of both, actually, for that. Here's another really extreme cancer phenotype here as well. Missing lots of autosomes, 1 copy of 2, 1 of 3, none of 14, etc. So these are really sort of classic things. They can be really quite diagnostic, and in other cases less so, but probably what's happening through the process. Yeah?
So why doesn't this just like render the cancer cell just like it can't do anything? Why is it still
How? So the question here is, why don't these really messed up karyotypes cause a huge problem with the cancer cell actually being able to replicate? Again, it's a great question. And in some cases, maybe these are dead ends. They're not actually going to replicate anymore. And one of the things that people are trying to do right now, karyotypes are a pretty gross way of looking at this. Again, you don't have much high resolution in terms of the genes, in terms of how these things are interacting with each other. So one of the things that people are doing right now are sequencing, getting the whole genome sequence of lots of cancer cells. So it's an ongoing project right now, and in fact, very heavily involved at OHSU, looking at now the genome sequences of all of these things and to try and get an idea what might be going on, how they can still replicate, why they will metastasize, so go from one place in the body to another, et cetera. And so they've got all these data, and now they're trying to understand how it's all fitting together in terms of how it works. But still, very powerful technique, this whole karyotyping thing. So that brings us to, hopefully, our last clicker question for today, which is how many pairs of autosomes are there in a normal human karyotype? 1, 2, 22, 23, or 46? Five, 3, okay, yes, normal human. Okay, we could argue whether males or females are normal, but autosomes, so it's not including the sex chromosomes. So 22 pairs of chromosomes. Okay, so let's talk about what's in a particular human chromosome. This is an example of one of the smallest human chromosomes. It's only 50 million base pairs in length. It was, in fact, the very first one to be sequenced. It's human chromosome 22. And in a metaphase chromosome, looks a lot like this. You start to zoom into that, and again, because of the genome sequence, we can now look at what's going on in each of these pieces, you'll notice that there's quite a few potential genes in here. All of these variously colored bars here are genes. You zoom in a little bit more, one of the things you see, there's actually a bunch of non-coding sequence in here, sort of what they call intergenic DNA. And we talked about this a little bit before. That's just outside of the genes, not introns and exons. We'll get to that in just a second. These are just extra pieces of DNA that are there. And we really don't know what most of these things are doing. Are they junk? Are they just left around? They use the analogy in the textbook of a really messy garage or fridge or life. Sounds like the last one sounds like me. So just a bunch of stuff sort of kind of thrown together. And whether that's been maintained historically and evolutionarily, or whether there actually are some things in there that we're not quite understanding what they are, that's still very much up in the air. One of the things also to mention with this chromosome, it's actually pretty tightly packed in terms of genes as compared to a lot of the other ones, which is also one of the reasons they started with it. Yeah?
Do the non-coding segments in a gene, is it possible for a coding strand to become a non-coding strand?
So we'll talk, so the question is, can coding become non-coding? We'll talk about that in just a second here. Actually, it's literally in the next slide. It's what people call pseudogenes. So yeah, we'll get to that in just a second. But in that particular segment, here we zoom in yet again in one of those genes. And genes here we're going to take as a protein coding gene. As I hopefully mentioned earlier on, genes can also be encoding RNAs, and we know a lot less about those genes that are coding for RNAs. A few of them we know really well, ribosomal RNA genes, tRNA genes, but there's lots of other RNAs that are present inside the cell. But in this particular, what's normally called a gene, you've got a number of regulatory DNA sequences, and then these little tiny red things here. And these little tiny red things here are the pieces of that DNA sequence which encode what's going to become your protein. So the actual amounts of protein coding sequences in the human genome is only about 1.5%. It's really, really tiny, the amount of actual protein coding genes that are in there. Lots and lots and lots of other DNA. Again, what it's doing there, it's the extra stuff that you've thrown onto your workbench in the garage. That will be me. Or is it something which is really important? I think the answer to that is we really don't know. And just a little bit more emphasis on how really, really big DNA is, and the chromatin problem that we have. If each base pair was 1 millimeter and you lined them all up next to each other, it would stretch all the way across Africa. That would be one human genome, 1 millimeter each for each of these things. This is ridiculously huge because of all this extra non-coding DNA. And why? Because it's there. Evolution just has to work. It doesn't have to be optimal. It just has to work. And so maybe that's the answer to it. So if you look at the human genome, again, we've got the whole sequence. It's getting better and better. We know more and more of the sequences which are there, but it's not the human genome. Well, we're not all identical to each other, are we? So there are all kinds of variations as far as that's concerned. Here, they say it's a 3.2 billion base pair genome. It's actually probably slightly bigger than that. Turns out it's actually a little hard to get the absolute exact number for these things, and I can show you some more of that a little bit later on. This is for the haploid genomes, just one copy. And usually 3 billion is a pretty good rule of thumb in terms of thinking about it. Number of protein coding genes. I love this approximately here. That's very much the case. This is about 21,000 genes. Remember how many genes there were in C. elegans? About 20,000. How many genes in Drosophila? About 20,000. We like to think of ourselves as more complicated than some of these organisms, but got about the same number of protein coding genes. One thing that is a little different in humans is that the size of these protein coding genes is a little bit bigger than many of the other ones. There's more of the introns, the non-protein coding sequences, than in a lot of these genes. Quite why that is again an open question that we don't quite understand. Exons, this is that protein coding portion of the gene. Some have one, some have a whole bunch. And it's usually about 10. So about 10 pieces of coding sequence that you have in any particular protein coding gene. These vary a whole bunch, particularly in terms of the largest ones. This is a ridiculously large exon. But a mean exon size is about 150 nucleotides. And this is getting a little ahead of ourselves, but hopefully everyone knows that the genetic code is a triplet code. And so 3 nucleotides for every single amino acid. So what does that mean in terms of number of amino acids that are usually coded for in any given exon? About 50. It turns out that 50 amino acids is usually just about enough to make one domain, one little folded piece of protein, which is going to have a particular structure. So think about moving exons around, maybe moving introns around, but particularly moving exons around, you'd be moving domains around. When we talk about protein structures, we said lots of these have different domains that are attached to them. So this would be a way, evolutionarily, if you move these pieces around, sort of a module effect. Turns out that's actually very true in terms of a lot of ways these things work. Number of non-coding RNA genes. This is, when they say approximately 9,000, that's when the textbook was written. It's probably now about twice that many, if not more. So there are probably more RNA coding genes than there are protein coding genes in our genome right now. Number of pseudogenes, getting back to your question. So a pseudogene is one that used to code for a protein, but now clearly has some major issues with that particular gene. And usually that's going to be some kind of nucleotide change which would clearly completely mess up the activity of that particular protein. And we'll talk a little bit more about that a little bit later on. I mentioned the 1.5% before. That's, again, pretty standard in terms of the protein coding sequences in the human genome. Most of our genome is in these repeated elements, and we'll talk much more about those repeated elements a little later on when we talk about transposons and jumping genes, jumping DNA. So I want to finish up just talking really briefly about chromatin. Talked about this before. We'll probably just get through nucleosomes and won't have a chance to talk too much about chromatin remodeling here. This is basically exactly the same thing that I showed you before. The most non-compressed DNA that you'll find inside a eukaryotic cell is one here with the nucleosomes, nucleosome, spacer, etc. Most of the time, these are packaged all together to form this. What each of these blobs look like? It's the nucleosome, which is 8 proteins that come together. Here, 8 histones. The H2A, H2B, which are here in yellow and red, respectively, H4 and H3, which are the blue and green. These all come together and wrap about 150 base pairs of DNA around this structure. So the nucleosomes are the DNA plus the protein. It's the histones that are on the inside. The DNA is wrapped around the outside. A couple of things I wanted to point out in terms of nucleosome, and we'll talk more about the nucleosome as we move forward on Monday here. But it's a very compact structure here in the middle. Not surprisingly, these proteins are extremely similar in all eukaryotic cells because they all have to do this basically same thing. But you've got the ends of DNA coming out at the top here, really quite close to each other. And this is really something that is very symmetrical. You look at it from the top, you look at the bottom. It's narrower here, and probably what happens in terms of that 30 nanometer filament is you end up stacking a lot of these things right next to each other. How do the proteins interact with the DNA? They interact in a sequence non-specific fashion. So sequence non-specific, is that going to be interacting with the major groove or the minor groove? Minor groove. It's going to be interacting mostly with bases or mostly with phosphates and sugars. Mostly phosphates and sugars, because that's what you find in all of these structures. So there are lots of negatively charged phosphates. What kind of side chains do you expect to see in your histones? Lysines and arginines, lots of positive side chains. Lots of hydrogen bonds, etc. We'll talk about how these things get put together on Monday. Have a good weekend and be safe.
for the next couple of days in particular.